Good afternoon, everyone. Welcome to our webinar series. The CSTFA is a research provider for sustainable tropical fisheries and aquaculture research globally. We provide world-class multidisciplinary solutions-focused research for Australian and international resource managers, both in government and the private sector. We're based at James Cook University in Townsville in far north Queensland, Australia. I encourage you to check out our website to find out more about us or send us an email. Today, it is my great pleasure to introduce Dr. Eva Sigsgaard. Eva is a molecular ecologist specialized in the analysis of environmental DNA or eDNA. Her main research interests are the mapping of and monitoring of biodiversity using eDNA and the possible uses of this approach in basic research, as well as in management and conservation. Throughout her research, Eva has explored the relationship between eDNA haplotype composition and true haplotype composition of a population in order to determine population genetic differentiation. Today, Eva will talk about the perspectives for population-level inferences from eDNA. She will present a brief overview of the current status of eDNA-based population genetics and outline the prospects for future developments in the field, taking into account the challenges associated with eDNA samples as opposed to those from individual tissue samples. Before I hand it over to Eva, I would like to remind the audience that there will be some time for Q&A at the end of the presentation. So you can just write your questions on the chat below at any time and we'll answer them later. So with that, I'll hand it over to you, Eva. Thank you very much.
Thanks a lot. So hello, everyone. Nice to virtually meet you. I hope I'll get to see some faces maybe at the end when we discuss a little bit. Yes, so I'd like to just start shortly with a bit of personal background since I don't know you. So I did most of my studies at Copenhagen University. So that looked mostly like this, you know, we don't have that much wild nature in Denmark. So kind of looking for the patches of wildlife between the farmland. But I was lucky enough to actually spend a semester at JCU during my undergrad studies. So I had a chance to do some field work where there were some more wild animals to see in the rainforest and in the mangroves. So during my master's, I started specializing in environmental DNA. And I did my PhD on how to use this method to investigate marine vertebrates. So as you may know, environmental DNA is, at least when we talk about macroorganisms, it's DNA that's left in the environment, left behind by different organisms. And that can be through for instance, shed skin cells or gut cells. And this environmental DNA can be used to detect species in a quite non-invasive and cost-efficient way. And in the aquatic environment, this has been done for more than 10 years now, both to detect single species and broader biodiversity. And this is usually done targeting short mitochondrial genetic markers. And this is because, eDNA is quite quickly degraded into fragments. So that's why we use short markers. And we use mitochondrial DNA partly in connection with this because we expect relatively low amounts of DNA. But mitochondrial DNA exists in many copies per cell, which gives us greater chances of catching it. And also there is quite a good reference, a lot of reference data. For instance, for the CO1 gene, we have a whole database just of CO1 sequences. You may know the bold barcode of life database. So, as I said, we can either do single species detection, and this is usually done using quantitative PCR, So here we find a marker that targets specifically a single species. So only the species should be amplified in the PCR. And then we can simply look at how much DNA is there and is amplified. But we can also look at many species at the same time. We can do this using metabarcoding. It's what we usually use. So in this case, we use PCR to amplify DNA or a short genetic marker from, for instance, all fish species. And then we compare these to a database, and we can find out which species were in an area. But as you all know, we are usually interested in more than just the presence of certain species. So for instance, there might be a native population of a species and a non-native population co-occurring in an area. So in 2016, there was a study by Uchi et al. Who used a similar method, or actually the same method as we use for single species detection, to distinguish between native and non-native populations of carp. And similarly, in 2016, I was involved in a study where we used metabarcoding, just targeting only a single species and using long barcodes compared to what is usually used. And in this way, we could actually identify different haplotypes of a whale shark aggregation. So most interestingly, we were able to compare the frequencies of these haplotypes with the same haplotype frequencies in tissue samples from the same aggregation. And this showed actually a remarkable correlation between these frequencies. And this is important because it gave us the ability to do some simple population genetic analyses just using the eDNA. So here you can see this is just a simple ordination plot comparing for this mitochondrial marker different whale shark populations around the world. And you can see that the eDNA samples here, they're quite close to the tissue samples. So we would be able to make similar inferences just from the eDNA. And also we were able to make an estimation of the effective population size of females, which was similar between the eDNA and our tissue samples. And it was also reasonable compared to previous estimates of the global female population. So there are many advantages and reasons why we use mitochondrial DNA. But since it's maternally inherited, the estimates that we get, they relate to the female history in the population. So for instance, as you saw, we could only estimate the effective population size of females. And for instance, sex bias dispersal could affect our results. Also, the mitochondrial genome is quite small and not that variable within species. So that gives us limited resolution. And because there's almost no or usually no recombination between the mitochondrial DNA and the nuclear genome, you could say it only counts as one single independent locus for genomic studies. So in a recent review, we looked at the future perspectives for population level inferences from eDNA. And here we focused on whether or how it might be possible to use nuclear DNA or nuclear eDNA for population studies. And especially if it would be possible to do genome-wide analyses. So I just wanted to make a quick stop here to hear if there are any clarifying questions. And you can just put them in the chat. So maybe we can at least start off with those later. Okay, I don't think I can see the chat actually. But I will just continue then.
Someone just raised.
Yeah?
Let me.
Okay.
I just saw someone raising their hand, but I cannot see that anymore. I cannot see that anymore. Maybe they changed their mind.
Okay.
Okay, maybe just
okay.
continue.
We'll take it at the end then. Okay. Great. All right. So usually for population genetic studies using nuclear DNA, in many cases at least we use restriction enzyme-based approaches, but these are quite indiscriminate. So they would basically cut DNA no matter the taxonomic source. I can actually see a question, I think. Okay, the distribution of females, how this can influence the estimate of total number. I'm not sure whether that would affect the population size estimate, but maybe for other analyses. So like, for instance, the population structure, if the females are actually moving differently. Yeah, maybe we can get back to that. So, so these, as I said, these restriction enzyme-based approaches, they're indiscriminate, and also they require large starting amounts of DNA. And we would not expect to have large amounts of nuclear eDNA. And of course, we would also expect to have DNA from several different organisms mixed. So we would want a more targeted approach. All right, so one possibility could be to do PCR targeting microsatellite regions or the flanks of transposable elements. And the latter is something that has been recently shown for ancient DNA. Regarding microsatellites, they can cause quite a lot of trouble with sequencing though, due to their repeating sequence and low GC content. Another possibility could be enrichment using hybridization capture. And you can see a little bit how this works here. So in this case, you design a lot of probes that are specific to the target organism. So probes are basically short synthetic oligonucleotides, a bit like primers. But in this case, you just basically pull down any sequences that, that match these probes. And then you can wash away any sequences that are not bound by these beads that we use. So Another challenge, which would make this different from when we use tissue samples, is that we wouldn't be able to link different SNPs, so SNPs from different sites across the genome. We wouldn't be able to tell which SNPs were from the same individual. We think this might become possible to some degree, at least, with linked read sequencing. So where you can track reads back to the original fragment. So if there was a long eDNA fragment to start with, you can kind of tag this with a molecular tag. And then the later reads that you get, you can track these back to the original DNA fragment. And then maybe at least assemble a few different SNP sites. And it might also be possible to do this with single cell sequencing, or perhaps based on read depths, different read depths obtained from different individuals. But until then, just having approximate allele frequencies could give us a lot of information. So from the previous studies on mtDNA, it looks like the frequencies of haplotypes actually match pretty well frequencies found in tissue samples. But of course, we cannot control the individual contributions to the eDNA pool, and they may vary for many different reasons, like for instance, the age of the individuals, which has been found to change eDNA production. Also, some allele frequency-based analyses are sensitive to rare haplotypes. And in this case, it might be a problem with some rare false positive haplotypes. And these can arise when you use PCR. PCR generates some erroneous sequences. And also, if we are using capture or PCR, we can get non-specific sequences, so sequences from other organisms that may lead us to think or to find haplotypes that are actually not true. So some possible solutions, sorry, it's very sensitive mouse. Some possible solutions for these problems could be to increase the number of individuals represented. So optimally, maybe this could be to target organisms when they are at highest densities. So for instance, when they aggregate, or some species if they make schools, or by increasing the sample size. Also, for tissue samples, recently there's been a lot of work on pooled sequencing, where you can save quite a lot of money, among other things, by pooling many tissue samples, and then simply looking at the overall allele frequencies. And we can use some of the statistical approaches from this method for eDNA as well, we believe, to estimate the different the sizes of the different variability sources. All right, and regarding the false positive haplotypes, there are different ways to remove them bioinformatically. So you can see here just a drawing of how you have the original sequences from different species, and then during PCR, there are errors that arise. There can also be chimeras, so sequences from different species can be connected. And during sequencing, there can also be errors arising. And then, of course, in order to avoid this unspecific capture or amplification, we would need to have reference DNA sequences from closely related species of the target species, so that we can actually design our systems or assays to be species specific. So as you can maybe see from all this, this approach would probably be most readily applied to aggregating or very abundant species where we can get many individuals covered in the samples. And also, if we want to make a genome-wide capture system, we would need preferably species with a reference genome, or at least a reference genome from a close relative. And to avoid this non-specific capture amplification, it would be best to have species either with no co-occurring close relatives, which can be a challenge, of course, or species where you have reference sequences for close relatives, so you can design the assay in a specific way. But if we solve some of these challenges, and at least for the species where it makes sense to use this method, it might be possible to get reasonable allele frequencies, which we can then use to, for instance, do mixed stock analysis, look at demographic history, and maybe even look at functional variation, which can then be compared between populations and maybe over time. And such studies looking over time are quite rare, as they take a lot of resources. And in this context, the eDNA method could have the advantage of being quite cost efficient. So just to say that even with just a single nuclear marker, we could make some additional inferences that cannot be made with mitochondrial DNA. Or maybe could be made in a better way. So if we can find a very variable marker in a species, then we could at least get like a minimum estimate of the census population size. And we could use rarefaction to estimate the total population size. Also, it could be possible to look at some sex markers, and then the read abundances might be used to look at sex ratios. It could also be possible to look at some epigenetic modifications. So some methylated, some markers that are methylated. And it has been shown that methylation patterns are affected by, for instance, aging and stress, at least in animals. So this could tell us about whether different populations differed in age and stress levels. And this can be detected using several different methods, including, for instance, nanopore sequencing. So it might be possible to detect this even in the field. And an issue with this is, of course, that methylation is tissue specific. So it would be necessary to, again, have quite extensive reference data, at least for those tissue types that we believe would likely occur in these environmental samples. So in water samples, probably gut and skin tissue types. And also, it might be possible to use eRNA to look at expression levels for some specific genes and compare between populations. So RNA has not been, or eRNA has not been used that much because it degrades typically very quickly. But on the other hand, this could give a very high spatiotemporal resolution. So eRNA would really reflect the exact time and space where it was found, even more than eDNA. And also, even though it degrades quickly, it has been found that organisms sometimes release quite large amounts of RNA. So it might actually be possible to have sufficient amounts to do these analyses. So just to recap quickly, at the moment, we're using only short mitochondrial markers to do eDNA population genetics. But it may be possible to do genome-wide nuclear analyses, or at least to gain some further inferences using single nuclear markers. And at least we speculate that it might even be possible to look at some epigenetic modifications, or to look at the differential gene expression between populations. All right, and then I would just like to thank my co-authors and our funders. And then I'd like to hear your questions.
Thanks, Eva. We have a question from Serena Adam.
Yeah.
Will pollutants and nutrients runoff affect the accuracy of the eDNA obtained? And can oxidants and denaturing agents in pollutants in low concentrations make a difference?
I mean, this is maybe, so I'm not a chemist, but so what I know is that the PCR can be inhibited by different substances. But usually what we have problems with is, so for instance, these humus substances from soil and substances from plants that can act as inhibitors. So I'm not really sure about these other, I don't think nutrients, I haven't heard that they affect the accuracy. But actually phosphorus affects how quickly bacteria they degrade eDNA, because if phosphorus is limited, then they might degrade DNA faster to obtain phosphorus. There was a study showing this. Yeah, I think that's what I can say.
Another question from Dean Jerry. I'll see if would you like to answer this live, Dean? Let's see if I can.
I can see the question as well.
Yes. Let me just give me a second. Dean, would you like to answer this question live? Or
oh, would I like to ask it? Sorry. Yes. Hi, Eva. Thanks for your talk. Very interesting. We've contemplated using eRNA as a way to detect the presence of parasites after a cleanup methodology that we would use. So we want to detect live parasites, whether they come through this process. And one thing we've looked at or considered was maybe RNA as the marker, because the DNA will still be probably present from the organism itself, whether it's dead or alive. Yeah. So my question is, like, are you aware of many studies of where eRNA is being used currently? And what do you think really are some of the technical challenges that we might have to consider besides the rapid degradation, I guess, of the RNA?
So I mostly know about some studies on sediment around oil platforms, where they use eRNA to detect species. And it looks like that it's true that it has more spatiotemporal resolution, so that it has a better correlation with environmental variables than eDNA. But that's just based on, I think, 1 or 2 studies. But there's quite a nice review about it. If you look at our paper, we refer to a review on eRNA. So I think that would be a good place to go. Yeah.
Yeah. Okay. Thank you.
I haven't used it myself yet. I mean, it takes you really need to to treat these samples properly. I mean, you need to get them, as far as I understand, you need to cool them down really quickly and be really careful with them to keep the eRNA intact.
Yeah, and preserve them in a way that you can deal with the RNA in terms of turn it into cDNA. Yeah, okay.
Exactly. And I think actually, like, the one might preclude the other, so you might have to choose whether you want to do eRNA or eDNA on the samples, or divide them up, because they need to be treated differently, I think.
Okay, thank you.
We have another question from Alessandro Ponzo. I'll allow Alessandro to talk so he can answer it. Ask it, sorry.
Hi,
sorry.
Thanks for the great talk. I was just wondering, based on your presentation, if, for example, looking at 2 populations of the same, or 2 aggregation of the same species, they are subject to different level of stress, assuming that the shedding of the tissues is relatively comparable. Do you think that we can use the level of methylation to assess the stress in these 2 different group of juvenile animal, right? So they're the same age, that should influence more or less the level of methylation, and they are the same species, assuming that the level of methylation from the digestive and the skin cells is the same. So do you think can be used with like nanopore to compare like the level of impact of human activities between 2 different aggregations, for example?
I mean, that's what we hope and speculate. I mean, as you say, but in this case, you would know that they were the same age. Well, I mean, I guess what we need in the best case, you would know that no matter the tissue type, the methylation pattern was the same, right? Because we wouldn't be exactly sure which tissue we were getting. But and I have to say, epigenetics is not what I work with, but if there are some sites that are methylated in the same way, regardless of tissue type, then I think that would be a very good starting point.
Thanks.
We have another question from Mari Cooper. I'll allow Mari to talk so she can ask it herself.
Thanks for your talk, Eva. I really enjoyed it, and your slides are really beautiful. Thank you so much. I was just curious if you had successfully isolated a single cell from an environmental sample, and if you had done this, if you had any like technical challenges, or if you had some things that you would urge people to consider if they were interested in doing this type of work.
I haven't done that. I know some people who have done it for algae from water samples, for plankton. But I know that it's possible to sort cells based on, for instance, the morphology and the cell size, and also the proteins on the surface. And that made us think that it might be possible to do at least some taxonomic sorting, which would, so in order to not get you know, to make it more efficient. So to get, for instance, perhaps we could get only vertebrate cells, or if that's not possible, at least only get the eukaryotic cells isolated. And then we could try to do some single cell sequencing. But if you're looking for a single species, I think it might be quite an inefficient approach.
Sure, great, thank you.
Any more questions? I think that's it.
All right.
Thanks very much,
a lot.
Eva. That was very interesting. And thanks for joining us, everyone. We'll see you next week on our webinar. Thank you. Bye.
Thanks. Bye.